Hi everyone, my name is Sofia Ramos and today I'm talking about crosstalk between expression of redox and calcium genes and its impact on breast cancer patient survival. The interactions between calcium and Roche signaling plays a crucial role in several cellular processes, including growth and cell death. Additionally, they influence each other. Calcium regulates Roche generation in mitochondria and cytosol, while Roche can impact calcium levels by regulating calcium channels, pumps, and stores. Cancer cells exploit this interaction to elevate Roche and calcium levels, which support tumor growth. These moderated levels are regulated by antioxidant enzymes and anti-apoptotic proteins. Breast cancer remains the most common cancer worldwide. Numerous studies have shown that dysregulated levels of calcium and Roche-related proteins are prevalent and affect cancer cell proliferation and migration. However, the mechanisms behind this dysregulation require further exploration. To address this, we asked, what if we combine the expression of redox and calcium-related genes to determine their impact on patient survival? Can they serve as biomarkers for breast cancer progression? Our aim is to define combined expression profiles of genes related to redox and calcium signaling and their association with breast cancer progression. To achieve our goal, we began by reviewing the literature to identify redox and calcium signaling genes associated with breast cancer progression. 39 redox signaling genes and 47 calcium signaling genes were identified. We then used two TCGA platforms, Walken to assess the statistical significance of differential expression between normal and tumor tissues, and GAPIA to investigate the impact of differential gene expression on breast cancer survival. We identified 9 redox signaling genes and 7 calcium signaling genes that met this criteria, and this is our sample. By collecting data from each patient using OncoLNC, we calculated the cumulative survival proportion at 5 years using Kaplan-Meier curves. We considered 4 scenarios, 2 associated with better prognosis and 2 associated with a worse prognosis. To identify genes with prognostic impact, we determined their association with biological processes through expression correlated genes and conducted an enrichment analysis. For easier interpretation, we categorized the processes based on shared gene pairs. Our final goal was to predict which gene pairs contribute to dysregulated biological processes associated with breast cancer progression. Our inclusion criteria allowed us to identify these 9 redox signaling genes and these 7 calcium signaling genes. These genes formed 4 possible combinations. In patients with a lower expression of one gene, if the increased expression of other gene increases survival, like this light blue curve, patients have a better prognosis. But in patients with this lower expression of one gene, if the increased expression of other gene decreases survival, like this light red, patients showed a worse prognosis. The other scenarios are these. Are related to high expression of the first gene. So, in patients with higher expression of one gene, if the increased expression of other gene are associated with an increase in survival, the patient have a better prognosis. And in the patients with higher expression of one gene, if the increased expression of other gene decreases survival, like this red line, patients showed a worse prognosis. We summarize these four possibilities in these four quadrants, and the upper quadrant are related to the left graph, and the downer are related to the right graph. But we asked also, in which process did genes or correlated genes are involved? The overlapping of correlated genes with selected genes show that most of these genes are involved in cell cycle regulation. As shown, these genes are upregulated in breast cancer patients, with many of them repeated in more than 3 gene pairs. Other gene pairs have correlated genes involved in adhesion and cell projection processes. However, in contrast to the previous group, most of these genes are downregulated in breast cancer patients. So, we can conclude that the simultaneous dysregulated expression of several gene pairs significantly impacts patient survival. The combination of high expression levels of most of the identified genes contributes to poor overall survival. Co-expressed genes are mainly related to cell cycle regulation and adhesion and cell projection processes. Validation of these findings with in vitro models can open new perspectives of understanding the molecular calcium and ROS mechanisms involved in breast cancer progression. Thank you for listening to me.